This is Dr. L, and we're going to talk now about hydrogen bonds. Note that you really can't understand this unless you understand some things about the properties of atoms, especially electronegativity, and also understand the difference between polar and nonpolar covalent bonds. If you don't get that stuff, you need to go back and review it and learn it before you move on to this, because this knowledge builds. So remember talking about polar and nonpolar covalent bonds. It's in a different video. Please review it if you don't. You can have, there are different types of covalent bonds, and one category is to be nonpolar versus polar. In nonpolar bonds, the shared electrons are shared equally, so there are no regions of partial charge, of obvious constant partial charge on the atoms in the molecule. These types of molecules do not participate in hydrogen bonding. Molecules that contain polar covalent bonds, like water molecules, that's a different story. These kinds of molecules can participate in hydrogen bonding. Polar covalent bonds means the shared electrons in the covalent bonds holding together all of the molecule or part of the molecule are not shared equally because some of the atoms have greater electronegativity than others. Here, oxygen is very electronegative, hydrogen is not that electronegative. The shared valence electrons are pulled towards the nucleus of the oxygen. This unequal distribution of these shared electrons leads to a partial negative charge on the oxygen and a partial positive charge on the hydrogen atoms. So this is a water molecule, and the colors are different, but it's showing the same ideas. It's showing polar covalent bonds. The shared electrons are pulled towards the oxygen, partial negative, partial positive. This structure is incredibly important, and it helps us to understand the properties of water, which is a very, very important molecule, because water is the universal solvent in which all biochemistry, all life occurs. It's essential to life, and it's like we definitely need water to have all of the properties that it does. Here are two water molecules in this B part of the figure, and we're seeing now our first hydrogen bond highlighted in yellow and shown as a dash. So here's one intact water molecule and a second intact water molecule. And what a hydrogen bond is a weak electrical interaction between an area of partial negative charge and an area of partial positive charge. This partially negative oxygen, this partially positive hydrogen atom are attracted to one another. This is a much weaker bond than a covalent bond. So when water molecules are next to one another in solution, these bonds will tend to form and break. But these are the bonds that will tend to hold these two molecules together. So this is intermolecular bonding in this case. Here we see the colors are different, but again, these are water molecules. So here's one water molecule, here's another water molecule. Each molecule is held together by polar covalent bonds. Each water molecule has areas of partial negative and area of partial positive charge. And the molecules, when next to one another in solution, like a glass of water or a lake or a pond or a water droplet falling from the sky, we'll see hydrogen bonds forming between areas of partial positive and area of partial negative charge. So here this leads us to the question, why does ice float? And this is actually a really interesting question because you probably take it for granted, like most of us do, that ice floats. But in fact, for most substances, the solid form is denser than the liquid form. And water is a little bit weird in that the solid form is less dense than the liquid form, ergo ice floats, like we see in this glass here. And this has to do with hydrogen bonding. So in liquid water, the hydrogen bonds between the molecules are forming and reforming, breaking and reforming. And they're just constantly doing that. In ice, however, what ends up happening is you get these incredibly stable, constant hydrogen bonds forming. And because of the 3D shape of water and the locations of the hydrogen bonds, all of these molecules get locked into a lattice-like configuration that is the ice crystal, and the molecules are actually a little bit further apart in this solid state than they are in the liquid state, and that makes solid water less dense than liquid water. And this is why ice floats. Hydrogen bonds are important important between some molecules like water, as we just saw, but they're also important within many molecules and many macromolecules. So this little ribbon-like structure it's a shape called an alpha helix, and this is a chain of amino acids, which is a peptide. And peptides, when they fold up, as this one is folding, become proteins. And so the peptide is formed when amino acids are covalently bonded together to make a protein. But hydrogen bonds are incredibly important too. So here we have a hydrogen bond, here we have a hydrogen bond, and over here we have a hydrogen bond. These hydrogen bonds form within the molecule, twisting it into this shape, which is called an alpha helix. And that's just one example of a protein three-dimensional shape that is gained by having intramolecular forces called hydrogen bonds. It's actually intramolecular. Intra means within the molecule, and inter means between two different molecules. So intermolecular, intramolecular. And hydrogen bonds are important in both. Hydrogen bonds are also incredibly important in how enzymes work. Enzymes are generally proteins, and they are catalysts for chemical reactions, absolutely essential in biochemistry. Enzymes they're like large, three-dimensional, generally protein structures, although sometimes RNA. They'll bind to a chemical, which we then refer to as a substrate, and it's shown in orange here. And then they'll change it into something else. So for example, the enzyme lactase will bind the substrate called lactose, which is milk sugar, and bust it in half, forming glucose and galactose, which we can digest more easily. And so the binding of the substrate to the protein enzyme happens at a particular location called the active site. We'll be learning about this in future classes. That binding is incredibly important and incredibly specific, and it is mediated by hydrogen bonding. So here's like a crude cross-section. The part of the enzyme that binds the substrate is called the active site, and it'll have parts of molecules pointing out that have little bits of partial charge, and the substrate will too, and hydrogen bonds will form between those areas, keeping the substrate attached to the enzyme long enough for the enzyme to do its work. All right, and that's enough about hydrogen bonds.